Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing Central University's Common Entrance Test, that is MSc Entrance question paper for the subject Plant Biotechnology, conducted in the year 15th June 2023. If you are new to my channel, please do like, share and subscribe. If you want still previous year question papers of CUET, you can refer to my channel playlist, that is go to channel Omni Bio Entrance, select channel playlist. From there you will see for search for CUET. In that folder you will find the question papers along with answers explained. Coming to the first question. The chlorophyll deficiency is seen for the seed viability, plant height, tillering and chlorophyll deficiency in the soma clonal variants in rice. That's correct. The plants obtained after soma clonal regeneration do not show variation. Okay. Now what is soma clonal variation? It is a genetic variation that occurs in plants which are regenerated from somatic cells through the method of tissue culture. So these variation, they result in changed morphology, altered biochemistry, anatomy, and these soma clonal variation, they can be due to genetic mutation, epigenetic changes, chromosomal rearrangements, deletions, etc. So plants obtained after soma clonal regeneration, they do show variations. So A here, A is correct but R is not correct. Answer is option 3. Which of the following is not a free living non-symbiotic bacterium? So Frankia, it is the answer. Frankia, it is a lives in symbiotic association with the plant, that is usually the actinorhizal plant, fixing nitrogen in exchange for carbohydrates. It is not a free living. The others are all free living non-symbiotic bacteria. So the answer here is option 4. Then next one. In potato, the selected protoclones display stable improvement of agronomic traits, which in turn include resistance to late blight. That's correct. Protoclones are genetically identical plants or cells derived from single progenitor cell through tissue culture. They are protoclones. That is, they are clone of cells. So in potato, the selected protoclones display stable improvement of agronomic traits, which in turn includes resistance to late blight is correct. The chromosomally normal plants with the characteristic different from the parental lines display an improvement of agronomic traits. That's also correct, but it doesn't have any relation with assertion. So A and R are correct, but R is not the correct explanation of A. Answer is option 2. Then next one. Virus-free plants propagated via tissue culture can be easily tested by. So it is ELISA, enzyme-linked immunosorbent assay, that is used. So ELISA detects specific viral antigens in plants extract using antibodies. So what you have to do is, first you have to prepare the plant extract, then coat the microtiter plate with antibodies specific to the target virus, then add the plant extract to the wells, incubate, then wash, then add enzyme conjugated antibodies, add substrate and measure the absorbance. If it is high absorbance, it indicates virus presence, and if it is low absorbance, it indicates virus absence. So that is why how ELISA is used in tissue culture for virus-free propagated plants. Then BioDart, an effective biopesticide, is targeted for the crop tea. Answer is option 2. Then next one. Trichoderma targets the pathogen Fusarium and could be used for the treatment of diseases like damping off. That is correct. Trichoderma reduces mycoparasites. Trichoderma acts by nutrient solubilization. Trichoderma acts by nutrient solubilization is incorrect. It is either either directly parasites and kill the other fungi, produces compounds inhibiting fungal growth, or compete with the other fungi for nutrient and space. That is how it kills the other fungus. So here statement 2 is not correct, but statement 1 is correct. So the answer here is option 3. It doesn't kill the fungus by nutrient solubilization. So answer is option 3. Next one. Two statements. Okay, there are two statements. One is right or the other is right or both are right. You have to find out which one. Statement 1. Protoplast are plant cells with intact cell wall. No, they are plant cells with their cell walls completely removed and are most commonly prepared from leaf. That portion is correct. Leaf mesophyll cells are usually used. Enzymatic isolation of protoplast is more suitable method over mechanical isolation because enzymatic protoplast isolation procedure yields less damaged, high quality and pure protoplast. That is correct. There are two types of protoplast isolation, that is enzymatic isolation and mechanical isolation. Enzymatic isolation is usually the one which is more preferred because it results in less damaged, high quality pure protoplast. Whereas mechanical isolation, it doesn't give, it results in more damage. So statement 1 is incorrect, but statement 2 is correct. So the answer here is option 4. Then next one. Heterocysts are specialized cells with thick cell walls without pigment system too complex. That's correct. Thick cell wall and no pigment system too complex prevents the oxygen diffusion into heterocyst, thus facilitate the creation of anoxygenic condition suitable for nitrogenous enzyme. That is also correct and that explains the assertion. So both A and R are correct and R is the correct explanation of A. Answer is option 1. Then next one. Trichoderma are mycofungicides. Mycofungicides means that they are fungi that kill or inhibit other fungi. They are mycofungicides. They lack the ability to survive under unfavorable conditions. They can survive under unfavorable condition, that is drought or temperature extremes. They are aggressive against phytopathogenic fungi. Yes, that's correct. They cannot utilize soil nutrients. They can utilize soil nutrients including organic matter and minerals. They are pathogenic to soil bacteria. They are not pathogenic to soil bacteria. But they can coexist with bacteria and it will help in the benefit of the bacteria. So the answer here is option 2. They are aggressive against phytopathogenic fungi. Answer is option 2. Then next one. Anther culture, pollen culture, though could be performed successfully but suffers from poor results and make it a lesser preferred method because of. So what are the disadvantages of anther culture and pollen culture? High degree of albino formation. Yes. Androgenic albinos show poor differentiation of grana. Yes. The 23S are totally lacking. Yes. The 16S are totally lacking. Yes. The protein 1 are totally lacking. Yes. So A, B, C, D, E answer is option 1. Next one. Contribution of Edward C. Cocking in the field of tissue culture, protoplast isolation. Answer is option 2, protoplast isolation in large scale. Then next one. Endangered plants. Endangered conserved by micropropagation. A3. Micropropagation technique, it is a product produces large number of plants. B4. Grafting and cutting, it is propagation methods. C2. Secondary metabolites, isolation of superior variant. D1. So the answer here is an option 2, A3, B4, C2, D1. Then next one, two statements are given. High number of transaminases are present in microorganisms. Yes. Glutamate dehydrogenase and transaminase work in cooperation under high ammonia concentration and assimilate ammonia in a variety of amino acids. That is also correct. So both statement 1 and 2 are correct. Then assertion, reason. Exogenous application of gibberellic acid inhibit the somatic embryogenesis of carrot in non-embryogenic callus. Yes. Gibberellic acid was isolated from filtrates of cultured fungus Gibberella fujikuri. Okay, that is also correct, but it doesn't explain the assertion. So A and R are correct, but R is not the correct explanation of A. Answer is option 2. Upon performing micropropagation in Jatropha curcas, it was observed that the callus would repeatedly become brown and die. Okay, so that is the this callus becoming browning and all is due to polyphenol secretions. What could be the possible reason? Explant secreted phenolic compounds. There was oxidation in explant tissues. Answer is option 3. Then next one. Ideal tissues for the plant tissue culture is apical meristem. Yes, of course, you will get virus-free plants. The viruses are unable to take over control of the host biosynthetic machinery. Auxin in meristem inhibit viruses. That's correct. So both A and R are correct, and R is the correct explanation of A. Answer is option 1. Abscisic acid antagonizes the growth promoted by gibberellic acid. Yes. Gibberellic acid promotes tertiary element differentiation in Jerusalem artichoke, while abscisic acid inhibits induction of tertiary element in callus cultures of Jerusalem artichoke. That statement also is correct. So both statement 1 and 2 are correct. Answer is option 1. Microbial application in agriculture involves. So it involves the use of bacteria, fungi, viruses as biopesticide and bioinsecticide. First option itself is correct. Answer is option 1. One of the statement about hydrogenase is not true. Bacteroids do not utilize hydrogenases, which is used for generating ATP. That itself is the answer. Bacteroids, they are symbiotic forms of rhizobia living within legume root nodules, and they do utilize hydrogenases to generate ATP. So first statement itself is the answer. Next one. The chromosomal elimination involves the endonuclease activation and genome specific fragmentation, which leads to chromosomal elimination. It involves endonuclease activation and genome specific fragmentation, which lead to removal of alien genome. Answer is programmed cell death doesn't involve chromosome elimination. Generation of small DNA Okazaki fragments also does not involve chromosomal elimination. DNA replication also does not involve chromosomal elimination. So answer here is option 2, removal of alien genome. Then next one. Induced fusion of freely isolated protoplast from different sources could be performed by. One statement is not correct. As the surface of isolated protoplast loses negative charge around the plasma membrane, which would otherwise repel each other. That itself is the correct answer. Answer is option 1. Induced fusion may be performed either in the presence of a suitable fusogen. That's correct. Fusogen may include sodium nitrate, yes, or treatment with calcium ions at high pH. That's also correct. So statement 1 is not the right answer. Then, in nature, haploidy is controlled by genes. Single gene called haploid inhibitor genes. Answer is option 1. Cry genes are for your Bacillus thuringiensis, HIV genes in HIV, multiple gene associated with HSP protein. HSP is heat shock protein. It doesn't have anything to do with haploidy. So the answer here is option 1. In vivo production of haploids cannot be performed by standard pollination method. This standard pollination method, it result in diploidy for offsprings, not haploidy ones. So answer here is option. Then crown gall A3, agrobacterium tumefaciens. If you know that, then option 1 is the right answer. In all the others, A is not 3. Okay, now bacteriocins B4, colicins and phage-like tail-like bacteriocins B4. Bacteriophage, it infects bacteria. C2, post-harvest loss control spores of antagonistic fungi. So answer here is option 1. Then assertion reason. After suitable treatment of plant tissue culture explants with cellulase and macrosome enzyme mixtures, so it is protoplast isolation. In that only you will use cellulase and macrosome enzymes. The protoplast are isolated and the whole suspension is floated onto 21% sucrose. Okay. And centrifuged to obtain a pure preparation of protoplast collected at the interface. That's 100% correct. 21% sucrose, in addition to providing a denser medium, also has higher osmoticum. This results in the preparation of high volume of protoplast. That is why you use 21% sucrose for protoplast isolation. So the answer here is both A and R are correct, and R is the correct explanation of A.